So welcome everyone. My name is Wilson. I am a postdoc at the California, San Francisco, and along with my University of Toronto colleague Shrey, we are delighted to have Dr. Ben Ouyang present for the first Zoom NanoBio Journal Club meeting this fall. And he will be speaking to us today about his recent Nature Materials paper entitled The Dose Threshold for Nanoparticle Tumor Delivery. And Ben has asked me to tell you guys, if you have any questions, please type it into the chat box, and then I will flag him down and he can answer them at the sort of in between different slides as he has the opportunity to do so. So take it away, Ben.
Wonderful. Thank you, Wilson and Shrey, for inviting me. I'm honored to be the very first speaker of the Fall 2020 Zoom NanoBio Journal Club. I'm really excited to share our paper with you today, the dose threshold for nanoparticle tumor delivery. And I'm really looking forward to the questions that you have and the discussions that we'll be able to engage in throughout the presentation and by the end of the presentation. Yeah, I wanted to try something new, make things a little bit more interactive. And so I wanted to start off by asking this question in a poll. Have you read this paper? And depending on the answers, I have 2 slide decks prepared. So depending on the answers, if people have read this paper already, I'll go through a little bit more of a more subtle slide deck with different things to discuss. So I'm going to launch the poll now. If you've read the paper, the definition of reading the paper is it can be, if you've downloaded the paper, you've read the abstract, and you've kind of scrolled through the main figures, that can count as reading the paper, right? So we're going to launch the poll now. And just by a simple yes or no, let us know if you've read the paper or not. And you should see a poll come up now and submit yes or no. Is this working?
Yes, it is working.
Nice, it is working. I am not seeing any responses on my end, though.
I see the responses.
Oh, you see the responses?
Yeah. Do you want me to end it? You've got 31 out of 33 people voted.
Okay, that's good enough. Yes, we will end it now.
And you should be able to see it now, I think.
And I'm going to share the results. Nice. Okay, so we've got about half and half. So what I'm going to do, that's more than I expected. So I'm going to then share with you the more, the different part of this then in a slightly different presentation. I'm going to share with you, there we go. Okay, I'm going to get back to screen share now. Slightly different presentation. We'll be able to discuss some of the subtleties, okay? And what I'm gonna do now is start a second poll. Okay, I just wanted to get a sense of the different techniques that everyone's using here. And I'm gonna ask the question, what nanobio variables do you use most in your lab? Right, so in the Chan lab, ever since we found the size mattered in the delivery of nanoparticles back in 2006, we do that for many of our studies now, so that's our favorite. But for you guys, what do you guys like? Let us know. The poll has been launched. And if there's anything that's here that, or if anything that's not here, let us know in the chat box of something that you prefer than the choices that we've given here. And again, Wilson, I can't see who's voted. So when you think it's done, then just then end the poll and then share the results.
Ben, are we supposed to be able to select more than one?
Yes.
We
Does that not work?
Or I can, yeah, it's not working.
Okay, if you cannot select more than one, then just select your most favorite one. You're still new to this polling thing here.
Okay.
I'll give them another 10 seconds.
Okay.
Great.
Okay, I'm going to end it now.
Cool. All right, so it looks like we've got size, surface chemistry, and material. And lots of people are using size. So this will be good. This is good because we have a little bit of an in-depth discussion about the concept of size here as it relates to dose. So I'm excited to be talking about this soon. All right, let's get this show on the road. So NanoBio. We have lots of different variables that we use to try and improve the delivery efficiency of our nanoparticles to tumors. But there is a little bit of room to improve. And what I'm going to show you now is the figure that we've all seen before. This is Stefan Wilhelm's paper back in 2016. And it essentially showed that over 10 years of delivery of papers that were published, we saw that there was a rather low amount of nanoparticles that were delivered to tumors. And importantly, that this trend had not been increasing over time, which kind of suggested to us as a field, we had not been unified on the nanoparticle delivery parameters that were important to delivering to tumors. And so we do have an arsenal of delivery parameters. We can do so many different things. We can change the material. We can use organic or inorganics. We can have targeting strategies, whether it's passive or active. We can change the size that many of us do here. We can change the charge, the shape, and we can even look at the biology to see if these things matter. And within each individual paper, there are remarkable improvement efficiencies that are done when we change these parameters. But as a field and as a whole, we haven't been able to suss out the specific thing that matters that can guide our design of nanoparticles clearly. And so what we were trying to do was then kind of find something that may be able to improve our nanoparticle delivery at a more general sense. And so we kind of isolated the biggest barrier to delivery. And for us, we looked at the liver, and we think the liver is the biggest barrier. You can see here that in an example of this, we have a picture of a mouse with a tumor over here on a flank, and we can see that nanoparticles deliver quite nicely into the tumor. But over here, overwhelmingly, we have the majority of nanoparticles that are stuck in the liver. And this is common through most of the nanoparticles, and it's actually quite joked around quite often that the definition of a nanoparticle is anything that accumulates inside the liver, right? And so this liver represents this major barrier to nanoparticle delivery, and it can take up to 99% of the nanoparticles. And it's really due to the macrophages that are inside the liver, right? These Kupffer cells that are inside the liver, they act as these filtration cells. They scrub the blood of any sort of nanoparticles that are in there and really get rid of these nanoparticles that accumulate. So I had three objectives for this more advanced presentation for those who have read the paper. I wanted to provide a rationale for a standardized dose metric, right? There are a lot of different ways of reporting dose in the literature right now, whether it's number of nanoparticles, whether it's surface area or mass. And I wanted to talk about why or which one would be most appropriate to be standardized. And how that kind of links into the mechanism of the dose phenomenon that we discovered, right? How does the liver actually clear these nanoparticles, and how do number of nanoparticles relate to that? And finally, to talk about the importance of dose in nanoparticle delivery as it relates to other studies and as it relates to humans. And so we'll start things off with our first objective. We'll talk about a standardized dose metric. And essentially what we had done was, I'll describe the experimental setup that was common throughout all of our, the majority of our studies here. We used 50 nanometer gold nanoparticles, and we also ranged them to 15s and 100s, so 3 different sizes. They were fully pegylated as much as we could, and about a fifth of these nanoparticles, a fifth of these pegs had a Cy5 marker on them, so that we could see them under fluorescence. Now the dose, we varied over 5 orders of magnitude, from 30 billion up to 550 trillion, because we felt that the dose itself, at some point, we would be able to overwhelm the liver. And we used 4T1 tumors in these BALB/C mice, and we had 2-week tumors that were roughly about 400 milligrams plus minus 50. And so this experiment was set up to look at the dose response in the tumor and also look at the dose response in the liver. And what we saw was extremely strong dose responses in each, right? So as we increase the delivery of our, or as we increase the dose of our nanoparticles from our lowest to our highest dose, what we saw was, we saw this delivery increase in the tumor. And we saw this inflection point that seemed to occur roughly around 1 trillion nanoparticles. And we saw a mirrored effect in the liver. The liver started to decrease at about 1 trillion, and it kept going down. And what I want to point out here is that the R squared value for both of these was 0.74 and 0.86, so relatively high. So suggesting that the dose was an important delivery factor for tumor delivery and for liver accumulation. And what I wanted to point out also was that to the right of each of these points is a number, and this number represents the size of our nanoparticles, right? So at the lowest dose, we have 100, we have 50s, and we hit 15s. And so these all kind of lie on the same line. Now, what we did was we normalized to dose here, and we used units of trillions just because that's where everything kind of lied in. But this is number of nanoparticles. And we asked the question, okay, is the number of nanoparticles the right way to normalize? What if we normalize to some of the other stuff that had been done in the literature? And so we said, okay, well, surface area is quite often normalized, normalization factor. What if we did that? And when we normalize the surface area, we see that this correlation starts to decrease in both the tumor and the liver. And interestingly, what we start to see is that we start to see these size-dependent effects emerge, right? So at this dose of 10 centimeters squared, what we saw was that roughly the hundreds had less delivery than the 50s, and the 50s had less delivery than the 15s. And this effect is mirrored in the liver, where hundreds tended to accumulate better than the 50s, and the 50s tended to accumulate better than the 15s, all roughly at the same dose of around 10 to 20 centimeters squared. Now, this started to show us an interesting question, right? If we normalize the surface area, then we can claim that there are size-dependent effects. We can say that smaller particles deliver better to tumors, and smaller particles generally avoid the liver. But maybe this starts to ask the question of a confounding variable, right? So whenever we normalize based on surface area, what we see is that we always give less large particles and more of the smaller particles, simply by the virtue of the normalization, right? Everything will be normalized by R squared. And so if we give more of the smaller particles, then that ends up with more delivery to the tumor and less accumulation in the liver. And this is also an issue when we normalize by mass. We see the same kind of thing. We see that the 15s deliver better than the hundreds. And this is common throughout a lot of the literature, where the conclusions have been that large particles don't really deliver that well, and they tend to accumulate in the liver, and the smaller particles do much better. So that kind of leaves us the question of this confounding variable, right? Which is the best way to normalize? Is it surface area? Is it mass? Or is it by number? And you can see that if we normalize everything by number, everything kind of lies on this line, with the exception of this weird ugly duckling point. But for the most part, especially in the liver, everything kind of lies on this line. And so then the question is when we normalize by number, is this the right way to do it because we saw a better correlation? Or is it the right way to do it because it has a much better biological implication as well? And so what I'll try to do now is to explain how the number of nanoparticles is a better fit by correlation and by biological and physiological explanation by telling you about the mechanism of this dose phenomenon. And so we can start by asking a very simple question. And the question is, how is this liver overwhelmed? Right? We know that by increasing the dose overwhelms the liver clearance, so less accumulates in the liver. This allows more nanoparticles to accumulate or to circulate in the blood. And the longer circulation allows more delivery to the tumor. So the question is, well, how is this liver being overwhelmed? And the first thing that we can ask is it cytotoxicity? You know, when we give super high doses of our gold nanoparticles, is it killing the cells that are related to the clearance? And that causes the increased circulation. And what we found was that the gold nanoparticles were not cytotoxic. We used flow cytometry and we used histology. And for the purposes of our presentation, I'll just show you a quick visual guide of this. Here we have immunohistochemistry, where we have macrophages and Kupffer cells in red. And what you can appreciate here is that at the low dose, at the low dose and at the high dose, there's no real difference between the numbers of the Kupffer cells in either of these conditions, suggesting that the Kupffer cells were not depleted. And this was also correlated on flow cytometry too. On the right, what we did was we used H&E staining to visualize some of the hepatocytes. And what you'll appreciate is that the hepatocytes also did not look that different between the low dose and the high dose conditions. one thing you'll notice here is that we have a lot of these black cells, and these black cells are essentially the cover cells that have taken up so many gold nanoparticles that they themselves have turned black. Right? But by and large, on both histology and the flow cytometry, we showed that the hepatocytes themselves are not damaged. Right? So we conclude that this inhibition of clearance was not due to cytotoxicity. So we started to ask, okay, it must be some sort of biological process then, right? Then we asked the question of, is it rate or is it capacity? And so it can only be these two different things. It's either that the rate is overwhelmed, and what that means is that there are so many nanoparticles flying past these cover cells that these cover cells cannot grab them out of the circulation fast enough, right? And they can't grab them fast enough, and more zoom by, and they continue circulating. The other question is it a capacity saturation? And for the capacity saturation, you can imagine the cover cell has eaten so many of these nanoparticles that they are no longer able to take up any more particles that pass them, and they're just too full to eat anymore. So we asked the question of, well, okay, is it a difference of rate versus capacity? And I see Wilson raising his hand here, and we have a question. We have two questions. Okay, so from Shrey, we have a question that asks, Ben, hundreds still do poorly at high doses. Do you think there's still a size threshold, irrespective of how you normalize? Yes, and this is a really important question, and I'll come back to that slide here. So there are two phenomenon happening here that I think is going on. If we look in the liver, there is a overwhelming of the liver accumulation. And you can see that these all lie on the line pretty well, regardless of the size, regardless of the 15s or the 100s, right? Even the 15s and the 100s accumulate in the liver at the same amount, regardless of the size, if you give them at the same dose, right? So I think in the liver, everything is governed by this one thing, of how the nanoparticles can bind to the liver. And I'll talk about that a little bit more as we talk about the mechanism. Over here in the tumor, we have this one weird ugly duckling point of the hundreds. And what this leads us to hypothesize then is that there is an additional mechanism for how nanoparticles accumulate in the tumor. And so for some reason, there is a size-dependent accumulation inside of the tumor itself, right? So there's one mechanism in the liver for the accumulation in the liver, and there's another mechanism for how it accumulates in the tumor. And there seems to be some sort of size-dependent mechanism, and we'll have to figure that out as we go on. Shrey, does that make sense? I see you clapping. So I'm gonna move on, and I'm gonna
I agree. I think like that's a very important point to point out, because like, yeah, we've also seen that the tumor has some sort of size thing going on, whereas the liver might just have that one factor that you're going to talk about.
Yeah. And I'll come back to this point a little bit later as we talk about the mechanism and why I think in the liver there is no real size-dependent effect. But in the tumor, we're still looking for that exact mechanism. All right. So Saad asks us, this cytotoxicity done only at 50 trillion, did you examine it with higher doses? As we now know the threshold, what is the upper limit? All right, so this cytotoxicity was done at a high dose of 50 trillion, I agree. I did not look at the cytotoxicity of the high dose that we looked at for 550 trillion. We didn't look at it in terms of flow cytometry, and we didn't look at it in terms of histology. What I can tell you is that at our highest dose of 550 trillion, we looked at the mice in general, and the mice did not exhibit any sort of toxicity symptoms. They did as they did, right? They were quite normal. They were happily running around. They were drinking water. They were eating. There was no real weight loss. So we don't believe that there would be any sort of cytotoxicity at that value either. Although you're right, we did not study it. Does that answer your question, Saad? I can't see his screen, so yes, Saad says yes. Cool, thanks. From Zara, she asked, when you evaluated the toxicity of the high dose, was it assessed at short term or at long term? This is a great question. For these sets of pictures here, I'm showing you this is toxicity at 24 hours after the injection, which is the typical time that we started to look at the biodistribution of our nanoparticles in the tumors and in the livers. We have experiments that are ongoing for long term. We have mice that have been injected with a high dose 50 trillion nanoparticles. They've been sitting there for about, I believe, 8 months now. And so we're going to assess at long term what happens at the toxicity. The mice seem to be quite happy. They're doing their thing, but we'll be sacrificing them in a couple of months, and we'll look at exactly what gold nanoparticles at a high dose does to these cells. So Alana asked, why can't it be both rate and capacity? What if rate is determined by capacity? This is a great question. I'm going to answer it as we get into the discussions. Mohammed asks, why did you choose gold nanoparticles? All right, this is a great question. I struggled with this question a lot for my PhD in the very beginning. So we chose gold nanoparticles because gold nanoparticles are very easy to detect. You can see that in this graph over here, we can very easily detect the amount of gold nanoparticles that make it into the tumor because gold is super sensitive using the technique that we have, which is ICP-MS. So we can detect up to sometimes even 1 nanoparticle in a sample, depending on the size. We can also synthesize our gold nanoparticles with high confidence and be able to characterize the dose that we gave. And so depending on their scattering properties, we can measure this on spectrometry. And depending on if we want to confirm it or not, we can also measure this on ICP-MS. Does that answer the question? Oh, and the other thing is that because ICP-MS is so sensitive over a very, very wide range of concentrations, so the ICP-MS allows us to detect over 7 orders of magnitude. We can also then vary our gold dose quite a bit to see the entire range of how it responds to differences in dose. Sweet, Mohammed says, thank you. Nice. Okay, Cecilia asks, when you say the cover cells look black because of nanoparticle uptake, did you stain it with silver staining? Oh, this is a really great question. So initially, when we were doing some of the low doses, we could only see our gold nanoparticles if we were to stain with silver. But what I'll mention here is that if you give a high enough dose, you will be able to see the gold nanoparticles without any sort of silver staining. This black is purely gold nanoparticles, no silver staining. And Wilson has answered, okay, nice. Nice. Kit asks, why don't you try the maximum tolerable dose? This is a question. This is a good question. We will continue to look at the dose toxicities. So for us, the majority of our studies were done at the 50 trillion dose, and so we understand what happens at that dose more comprehensively. So this is why we tested that. But you're totally right, and at some point we will try the maximum tolerable dose as well. The accumulation in the spleen. So Rajendra asks, the accumulation in the spleen along with the liver. This is a great question, and it's a question that we don't fully understand yet. So when we looked at the accumulation in the spleen across our doses, we saw that it flatlined. So there was no real dose dependency of the spleen. And it always seemed to flatline around 3% of the injected dose, whether we gave 50 billion or whether we gave 50 trillion. We don't fully understand how that happens. There are 2 different possibilities of how that could be happening. The one is that we never fully saturated the spleen, and the spleen is able to take up more and more and more. And so you can see that whatever flows to the spleen, it just takes up a certain amount. The other idea is that we have saturated some phenomenon in the spleen, and now it is limited to some fundamental rate of uptake, and it can only take up a certain amount. There should be studies ongoing that figure out what's going on in the spleen, but for now, we kind of skipped over it because we found that the dose dependency was really dependent on the liver and the tumor, and they almost seem to be this like opposite seesaw effect, where if you decrease the delivery in the liver, then you get this increased delivery in the tumor. So we really focused on that. But you're right, the spleen is a really, really weird one, and we want to figure out what's going on there. Yingyu asks, hi Ben, do you think dose will affect the margination of nanoparticles in the blood vessel and therefore affect the interaction with the cover cells? Yeah, this is a really good question. And what we found is that there was this margination effect. And what I'm gonna do is maybe I can even show you this. kind of an interesting margination effect here. In our paper here, we tried to look at this margination effect in vivo using histology. And so we gave two different doses, 0.8 trillion and 50 trillion. And what we did here was we looked at the distribution of gold nanoparticles at 15 minutes after the injection. And so you can see that when we gave a low dose of nanoparticles, they seem to be more distributed along the wall. Whereas when we gave a higher dose, more nanoparticles were inside the lumen. So we represent this graphically like this. And when we do the quantification again, so 0.8 trillion, they seem to be more along the wall rather than lumen. And if you give more, they end up near the lumen. And this kind of gives us a hypothesis that, you know, these nanoparticles, if they can not be along the wall, they wouldn't really be binding to any of the cover cells that are there. And the ones in the center, if they're not really touching any sort of cover cells, they have a higher probability of exiting that sinusoid. And if they have a higher probability of exiting in sinusoid, they have a higher probability of circulating, and that increases the circulation time. So, Yingyu, does that kind of answer your question? Go back to, nice, Yingyu says great. Okay, going back to the screen share here. I'm gonna screen share the PowerPoint. All right, so we're going back to the question of rate versus capacity. And Alana, I'll try to answer that question for you here. So before we can answer this question of the rate versus the capacity, we wanted to ask, okay, is it really the cover cells, right? And what we did was we, to prove that it was the cover cells, if we eliminated the cover cells, then there should be no dose dependency, right? And so what we did was then we first injected clonogenic liposomes, and we depleted the cover cells over 48 hours. Then we injected our gold nanoparticles. We waited 48 hours for these nanoparticles to distribute in the liver and then in the tumor, and we measured. And exactly what we found was that if you deplete the cover cells, then there is no difference in tumor delivery between the low dose and the high dose, right? Which really, really clenched our idea that the cover cells are really responsible for this dose dependency, okay? Now, if we know that the cover cells are responsible for the dose dependency, then we can more clearly look at the cover cells and see what's going on within them, because they're the ones that are governing this phenomenon. And so what we did here was we looked at the cover cell uptake rates, and we studied the uptake rates using intravital microscopy. And the reason why we used intravital microscopy was because intravital microscopy gave us temporal resolution down to the second. And that allowed us to assess the amount of nanoparticles that were taken up very, very clearly inside, in situ, in their native phenomenon, in their native environment, and at a single cell level. And so what you can see here is that we broke it down into two doses, a low dose and a high dose, and we studied the uptake rates. And in the low dose, we injected a sub-threshold dose of Cy3 nanoparticles, and our cover cell, a single cover cell here, is outlined in blue. And what you'll appreciate is over the course of these 25 minutes, this cover cell has taken up a considerable amount of these nanoparticles. Next, what we did was we compared it to a mouse that was injected with a high dose of nanoparticles. And in this high dose, we kept the same dose of Cy3 nanoparticles, and we supplemented with a above threshold dose of Cy5 nanoparticles. And this meant that when we observed in the Cy3 channel, we wouldn't see the uptake of the Cy5 nanoparticles. But what you'll appreciate is that over the course of these 25 minutes, there isn't really that much uptake. And there's just a minor amount of uptake inside this cover cell, which really suggested to us that there was some sort of competitive inhibition going on. These 12 trillion Cy5 nanoparticles were competing against the Cy3 nanoparticles and preventing their uptake, which really tells us that, okay, there's something happening that's impeding the uptake rate of these nanoparticles at high doses. And so Alana kind of asked us the question, can it be saturation of uptake rate and uptake capacity, right? And so here what we did was to be able to look at the uptake capacity, we visualized in the Cy5 channel. And we asked the question, you know, maybe it was saturated in capacity in the first 30 seconds. And if it was saturated in the first 30 seconds, then we wouldn't be able to have any sort of uptake, and we would see the same kind of effect. But what we see is that in the Cy5 channel, these green nanoparticles were continuously taken up by these macrophages up to 25 minutes, and even longer when we extend it to an hour. And so this really suggested to us that these nanoparticles were not saturating the capacity of these macrophages. And to kind of look into this more at a super resolution level, we also looked at it under TEM. And so this is the sinusoid of the liver, where this is a hepatocyte, and a hepatocyte. And this is generally the sinusoid of the liver. And you can see that there are red blood cells and a Kupffer cell in this sinusoid. And you can appreciate that this is a time point that's 15 minutes after injection of a high dose. You can see that these nanoparticles are inside the Kupffer cells, but they don't represent more than maybe 5% of the Kupffer cell volume. And compare this to a Kupffer cell that has been completely overwhelmed by nanoparticles that looks like this. So this is a macrophage that was cultured in vitro with nanoparticles. And I'm outlining it here. And we gave a ton of nanoparticles over 24 hours for this one. And you can appreciate that these nanoparticles take up maybe about 60 to 70% of the volume of the macrophage. And this tells us that at our 15-minute time point, there's no way that these Kupffer cell capacities have been overwhelmed. And so we wanted to ask the question, okay, well, you know, if it's the rate that's being overwhelmed, how is the rate being overwhelmed? And what I hope you'll appreciate is that all of these nanoparticles seem to be outlining the perimeter of the endosome of this Kupffer cell. Now, that makes us think that it's a binding site saturation, that these nanoparticles are binding to the membrane and they're saturating all of the sites that are available for binding on the membrane. And this is different than another hypothesis, which would be a macropinocytosis. And you can imagine if the Kupffer cell is non-specifically engulfing things with a macropinocytosis phenomenon, they're just kind of like waving their arms in the air and grabbing a bunch of nanoparticles. You would imagine that there would be particles on the edge, but there would also be particles in the lumen. But we never see that. We always see the particles on the edge, right? And this was common throughout all of the macrophages that we had seen at these short time points. And what I can hope that you'll appreciate is that they seem to take up all of the entire perimeter. There isn't that much space left for these nanoparticles to bind. And what that says to us is that there is some receptor on the surface of these Kupffer cell membranes that binds to a single nanoparticle. And as that nanoparticle binds, then that allows it to be internalized. And so if we have saturated all the room left on the membrane, then there's no more room for nanoparticles to bind. And if the nanoparticles can't bind, they just continue to flow past, and this allows them to continue to circulate. And so you can kind of start to see why we think that the dose by number is the thing that is more, is the dose metric that is the most appropriate in this situation, right? Because there will always be a fixed ratio of number of receptors to number of nanoparticles, right? Whether it's 1-to-1 or whether it's 2-to-1 or 10-to-1, there will always be that fixed ratio. And that ratio can be looked at if we dose by the number of nanoparticles. If we were to dose by the surface area of nanoparticles, that may also be true, but you can appreciate that within this nanoparticle, the only surface that's kind of binding to this membrane is this surface area here. And the surface area that's all the way out here, all of the surface area that's not bound to the membrane is kind of irrelevant and unused. And in terms of the mass, it's really only the perimeter that's binding. The mass itself, this particle can be really big or can be really small, but the stuff that's on the inside doesn't really relate to how it can be taken up by the liver. And so we really think that because of this correlation between receptors and the nanoparticles, we think that the nanoparticle dose by number is the most appropriate dose metric to use going forwards. And so I said that it wasn't macropinocytosis. What I'm going to show you here is another experiment that we did to confirm that it wasn't macropinocytosis. And what we did was we used 70 kilodalton dextran. 70 kilodalton dextran is a well-established marker of macropinocytosis. And what we did was we co-injected with our Cy3 labeled gold nanoparticles. And our hypothesis was that knowing that this dextran goes through macropinocytosis, if the gold nanoparticles and the dextran co-localized, then that would mean that the nanoparticles had gone through the same uptake pathway. And what you'll appreciate here is that we used intravital microscopy again, and we looked at the distributions within single macrophages up to an hour after injection. And what you'll appreciate is that there's a lot of stuff that's just red, and there's a lot of stuff that's just green, and that there isn't too much yellow, which really told us that the gold nanoparticles, again, confirming that they weren't going through micropinocytosis, they were going through this binding site mechanism that we hypothesized. Now, I wanted to ask the question then. For us, we think that it's some sort of receptor that must be involved, right? And so what I'd like to do now is to ask a question. What receptors do you think are involved in this binding site clearance from nanoparticles from the liver? I'm going to launch the poll now, and I'll give everyone some time to answer. And if you think that there are receptors that are outside of what we've listed, please type them in the chat. And while people are answering this poll, I'm going to answer some of the questions that have popped up in the chat. So Shrey asked, do you think when we do experiments in vitro, we are observing macropinocytosis versus when you do it in vivo, it's being receptor mediated? Or is it something that's related to raw versus cover cells? And Shrey, I'm going to ask you to clarify that question. What do you mean by this? Why do you think that one is different in vitro versus in vivo?
So what I was asking was, you showed two TM images. On the right, we actually saw the endosomes being full of nanoparticles, versus on the in vivo TM, they were lined up against the surface. So I was wondering whether, like, why don't we see that kind of image in raw cells?
Yeah, this is a really good question, Shrey. I'm going to try and find an example for you. If I show you, so the difference between the lining the endosomes versus being inside the endosomes is really a function of the time. And I think it's a function of the time biologically, because these endosomes will later fuse at longer time points. The nanoparticles kind of come off the receptors and they just kind of fill the inside. And so the image on the right that I showed, which I'll go to right here, the image that I show you here on the right, this is at 24 hours after exposure to nanoparticles, versus on the left here, this is 15 minutes after exposure to nanoparticles. And so you can see at the short time points, they line, and at long time points, they're on the edge, they're on the inside. And I can confirm to you that this also occurs in vivo as well. If you look at in vivo long time points, so this is 24 hours after the injection, they also end up in the center. And I can also show you that in vitro, I don't have that slide actually. So I do have TMs that in vitro at short time points, we also see that they line the edges. So this is not specifically an in vitro versus in vivo phenomenon. This is specifically a time-based phenomenon. So at short time points, they're going to line the surface. At long time points, they're going to be on the outside.
So the follow-up then is that, do we have both of them present and they're like differentiated by time, or is it receptor, like they uncouple from the receptors and then more load in or something?
Yeah, that's what I'm going to believe as well. They're unloaded over time, and there very well could be some nanoparticles that are internalized by macropinocytosis, even at short time points, right? You can see kind of here that there are these endosomes that have nanoparticles also in the lumen. And so, yeah, I wouldn't put it past it to believe that these macrophages would eventually just like reach up randomly while they do macropinocytosis, and at high doses, nanoparticles would just get taken up. So I agree that probably does happen. But overwhelmingly, what we see is that the majority of the nanoparticles line the outside edges. So we do believe that the main mode of internalization is through this receptor mediated.
Last small question. So, you know, when we were discussing the first thing where we were saying that liver might have only one thing and all the, like the size doesn't really matter that much. Mm-hmm. So can we extend that to saying that even for uptake mechanisms for liver, irrespective of what size you have, it will always be receptor mediated?
I think for what we've seen here, that seems to be generally the case.
So you saw this kind of uptake pattern for 15s and 100s as well, like lining up around the endosomal membrane?
No, so I didn't look at 15 minutes for 15s, but based on our ICP data and based on how it accumulated in the blood as well, we believe that this is how it would be taken up. But that would be an interesting experiment to do, to see if we did do the 15s at these short time points, we would also see it bound onto these membranes. Yeah, that's good.
That would also kind of tell us whether there is a common element in the corona, maybe across different time, like different sizes, that is mediating receptor. Like, I don't know. Anyways, that's another PhD.
Yeah, no, that's a good point. That's a good point. Good question. So we got Mohammed asking, does that mean that at higher doses, liver function, particularly Kupffer cell phagocytic and filtration function, is compromised? Would these mice's livers be more likely to let pathogens skip through? This is a really good question, Mohammed. First off, if it does compromise it, we have the majority of this clearance occurring in the first, let's say, roughly about 24 to 48 hours. And so if it does compromise it, it would be only a transient compromisation. And secondly, we found that there is a specificity for the phagocytosis in the liver. We found that even among different nanoparticle types, they seem to go to different cells, and one nanoparticle cannot enable the saturation of another nanoparticle. And what do I mean by this? So there was an experiment that we did that's in the paper where we injected gold nanoparticles at a low dose, so below threshold, and we co-injected with liposomes at a super high dose. And what we found was that the distribution of gold nanoparticles did not depend on whether or not there were co-injected liposomes, right? Which suggested to us that these liposomes could not saturate the binding of gold. So we don't have the exact experiment to show this, but this would also then suggest that potentially the gold nanoparticles would not inhibit the uptake of bacteria, because there is some sort of specificity. And the reason why we think this is the case, we did some protein corona studies to show this, is that the coronas of gold nanoparticles and the coronas of liposomes are different. And there's been some evidence in the literature to show that there are different Kupffer cells that bind different nanoparticles. And so for a long time, we thought that the Kupffer cells are this homogeneous population of cells that just take up anything that comes into their contact. But when we looked at the distribution of liposomes and gold nanoparticles when they were co-injected, it turns out that they went to completely different cells. And I'm going to show you what that looks like here. And you can see here, what we did was we co-injected these Cy3 liposomes with Cy5 gold nanoparticles. And what you'll appreciate is that liposomes and gold nanoparticles went to completely different cells. And some cells only took up gold nanoparticles without any sort of liposome uptake, right? Which might suggest to us that there are Kupffer cells that would only take up the gold and would not take up the bacteria. But that, to completely prove that, we would have to do that experiment, which we did not have the time to do here. But good question. Alana is asking the question, Shrey, I have been wondering about the in vitro stuff as well. I also think the receptor expression on these cells when they stick to a dish is different than in vivo. There are some papers that look at virus uptake that examine this. Cool. Yeah, Alana, we can have further discussions on this. Michael Trin asks, did you try to see if this endosomal uptake common across different surface chemistries? Uptake is common. I hope I answered that question with the liposome versus the gold. Rajendra Prasad asks, what is the mode of injection? The mode of injection across all of our studies was intravenous. So we did a tail vein injection. So we heated the mice up for maybe 15 minutes under a heat lamp, and then we injected into the tail. So Hong Lu asks, not in the nanoparticle field here, but I was wondering if the receptor in Kupffer cells that bind to the nanoparticles has been identified. Ooh. If so, would changing the receptor properties or changing the charge of nanoparticles be feasible strategies to further increase the capacity/rate of Kupffer cells? So this is what I wanted to ask in this poll. Have they been identified? What do you guys think it is? So I'm going to end the poll now. I think Wilson, most people will probably have answered by now. I'm going to end it. And I'm going to share the results. And we've got this distribution. So most people believe that it's Fc receptors, complement receptors, and scavenger receptors. And we've got a couple of thoughts on the LDL receptors, the antiphagocytic receptors that are a little bit inhibitory, such as the CD47 SIRP alpha axis. We got some of the inhibitory Fc receptors, and we got some of the others. Some people that have said others. Now, okay, and I totally agree with you. There's quite a spread of votes here, and I think it's because it's hard to identify which one it is. Right? We have some hypotheses on what this is, and we're going to figure this out, and we'll be able to hopefully identify them. And as you said, if we can identify them, then we can design things that avoid them, and then we would be able to avoid the liver clearance. And if we did that, then potentially we could change this threshold, right? There would be a new threshold that potentially would be lower if you had a nanoparticle that avoided receptor binding in the liver. And I hope that makes sense. Saad says, I think the in vitro is not a good predictor for in vivo. Yeah, I totally agree with you. This is why we spent so much time doing intravital studies, because we really wanted to look at the behavior of the Kupffer cells in their native environment. Super, super difficult experiments, but were very important for us to figure out the exact behavior. Kit asks, for repeated dose, I can see around 70% of nanoparticles are accumulated in the liver for the low dose. What will happen to the high dose after 24 hours? For example, 7 days. Will there be more nanoparticles accumulated in the liver? How will they be there finally? Will this number be similar to repeated injection? So Kit, I see that you've read the paper. I'm going to come back to that question because I'll present that slide in a second. Ying Yu asks, hi Ben, long time in vivo EM images seem like nanoparticles are in lysosomes. Yes, I totally agree. They're in late endosomes / lysosomes. Ben, just to refresh my memory, you only looked at one location liver for intravital. We looked at multiple intravital locations in the liver. I'm not quite sure what you're asking here.
Yeah, sorry, there's a typo. It should be like in the liver. Did you look at multiple locations or just one location?
So within the liver, what we tried to do for this intravital was we tried to focus at the periportal macrophages, because we know that the periportal macrophages are the ones that are the most phagocytic for our gold nanoparticles. And it's possible that outside the periportal, maybe in the perivenous area, they may have a slightly different behavior that we've seen in the past. But yeah, you're right. We looked, majority of our images were looking at the periportal region. Sweet. Okay, we are going to move on here at the receptors. I'm going to go back to the PowerPoint. We're talking about receptors. Just pull out of the way. Okay. So, yeah, most people thought it was Fc, complement, and scavengers, and we totally agreed. We're on our way to identifying these receptors. And so now we're going to go to the question that I believe Kit asked, which is the repeated versus the single dosing. And again, this is a manifestation of what we thought could be an outcome of the discussion of the rate versus capacity. Right? So if we know that it's the uptake rate that's being overwhelmed, then that means that we would see the best delivery if we were to maximize the concentration in the blood at any given time, right? So you maximize the concentration at any given time, then the Kupffer cells aren't able to take things up fast enough, and you get less accumulation in the liver, better delivery to the tumor. So we tested this idea by taking a single dose of 5 trillion nanoparticles, so something above the threshold that we have found, and we divided it into 8 different doses, 8 smaller doses that were all sub-threshold, right? So 0.6 trillion. When you sum these all up, the 2 doses were the exact same, about 5 trillion. It was just that for the repeated doses, what we did was we gave them once a day for 8 days, right? And we compared this to a single dose. And then at the end, we sacrificed the animals. And what we found here was very, very striking. So you can see just by eye that if you give the repeat dose, the tumors maybe they have a little bit of nanoparticles in them. But if you give the single dose, that you can see by eye that the nanoparticles are much better delivered to the tumor. And keep in mind that this was the same total dose that we injected, right? The only thing that we changed was the dosing regimen, which really told us that, when we measured on ICP, this confirmed that the single dose to maximize the amount that was in the blood is the best way to get nanoparticles into the tumor. And this correlated with all of the other parameters that we had measured too. So when we measured the half-life of these nanoparticles, the single dose maximized the half-life. And when we looked at the liver, there was much less accumulation inside the liver if you give a single dose. And I remember Kit had asked us the question. He said that 70% was in there at the repeated dose, and about 30 to 40% was there at the single dose. And he asked, what happens to the high dose after 24 hours in 7 days? So if we were to give the high dose, the single dose after 7 days and kind of just measured, waited 7 days and measured out here, what would that accumulation be? And I think it would not be too different because essentially what ends up happening is that this single dose, by the time we've gotten to 24 hours, this 5 trillion has mostly cleared from the blood. And so once the nanoparticles had cleared from the blood, they will have settled in their final organ sites and they don't move around too much after that. And so if we extended this to another 7 days and then measured here, there would not be too much difference, I would expect. And I hope that answers your question. We got another question here. Ben, did you check the stability of dye-conjugated nanoparticles against glutathione? This is a great question. We had a really interesting discussion of glutathione and how it works. And I guess Yingyu, you come from Ji Zhang's lab, and you guys have done excellent characterizations on how glutathione works in the liver. We had done initial experiments to characterize the stability of dye-conjugated gold nanoparticles against glutathione, but only in vitro. And so we incubated gold nanoparticles with a really, really high concentration of glutathione. I think it was like 10 millimolar or something like that. And we found that it took 24 hours at 37, something like that, for the dye to start falling off. Right. So we think that it's rather stable, but we're not quite sure how this works in the liver situation. And we were unable to do that. But if we followed that Nature Nano paper from your lab, I think we may be able to have a better answer. But it's a good question. It's a good question, and it should be an ongoing question for us later on.
Yeah, but this is a nice answer. I guess it also depends on the surface density of your PAC ligands. See what I mean? Yeah. Because if you have too many exposed gold atoms in zero valence state, then GSH will not etch the nanoparticles. It will just bind to the gold zero on the surface of your nanoparticles.
Yeah, I think that's a great point. The way we had done it was we had maximized the amount of PEG that was on our gold. So we saturated that surface. So roughly we get about 1 or 2 PEG per nanometer squared. The brush layer is quite thick and quite long. So this could be a potential explanation for why it took so long for the PEG to fall off of our gold under glutathione. Right? It's just hard to get in there. Yeah, good point. Thanks.
All right.
Okay. And I want to continue the discussion of the rate versus capacity, but first I wanted to just to kind of explain how this might work in treating nanoparticles, in treating tumors. Right, so we used a lot of gold. Gold doesn't really do that much. And in the paper, what we showed was that we can actually use a bunch of empty delivery vehicles to supplement liposomal doxorubicin. So it's Calyx in Canada. In the US, it's called Doxil. In Europe, it's also called Calyx. So liposomal doxorubicin, we made a bunch of empty liposomes without doxorubicin, and we showed that just by boosting the dose of nanoparticle without boosting the dose of drug, we could get an increase in the efficacy of Calyx. So you'll see here that these 2 gray lines represent a sugar control and the delivery enhancers on their own. So there was no difference between these two, suggesting that the delivery enhancers had no therapeutic efficacy on their own. When we added the Calyx, you can see that there was a little bit of tumor shrinkage. Now, when we co-injected the calyx with the empty delivery enhancers, we saw an additional 60% reduction in the tumor volume, which really suggested to us that these delivery enhancers that had nothing to do except distract the liver, they can be used to increase the efficacy of therapeutic nanoparticles. Right? And we also saw a slight increase in the survival as well. Now, I tell you that because now I'm going to go back to the rate versus the capacity saturation. And the idea here is that the co-injection should be better than sequential injection if it is a rate mechanism. And so what is sequential injection? So there's a technique out there that exists. It's called RES blockade. And essentially what they do is that they try to overwhelm the liver by first injecting a dose of the empty nanoparticles and then waiting some time for these cover cells to saturate their capacity. And then they inject a dose of therapeutic nanoparticles. And because the capacity of the cover cells had been saturated, the therapeutic nanoparticles would now whiz by the liver, avoid clearance, and be able to accumulate in the tumor much better. So we wanted to test this hypothesis, right? If it's an uptake rate thing, then the sequential injection should not be needed. So we directly compared our co-injection versus the sequential injection of the RES blockade. And what we found was that as we measured some of the secondary effects, we found that the co-injection if you look in the blood, the co-injection maximized the amount that was remaining in the blood after 24 hours, better than if you were to do a sequential injection and introduced a delay between these two injections. And especially as you increased the amount of time of the delay, the amount that was remaining in the blood decreased, which suggested that the longer you delay, the less effective those empty nanoparticles are. And of course, this kind of a half-life decrease also corresponded to a decrease in the delivery efficiency to the tumor. And when we look at the final efficacy of this, we see a modest but statistically significant effect of the co-injection. So the co-injection over here decreased tumors by up to 30% more than if we were to give a sequential injection over 24 hours, which really suggested to us then that the RES blockade, it's a good way to improve the efficacy of therapeutic nanoparticles. But really, what you should be doing is you should be injecting these nanoparticles together in a co-injection rather than doing sequential injection. There's a lot of other advantages to doing this too, right? Because if you're doing a co-injection, that means you've reduced the amount of deliveries from two separate injections down into a single injection. So it's practically easier. And if you were to translate this into patients, you would be making their visit to the chemotherapy ward a lot easier if you're just going to give them a single administration. Secondly, you don't have to figure out how to optimize the time delay between your first and second injections, right? Because here you just give a co-injection. There is no figuring out if a 15-minute delay is better than a 1 hour, is better than a 6 hours, better than a 24 hour. You just give them together. Really, really nice, really simple. And so I hope I've convinced you that the mechanism of the dose phenomenon is based on this overwhelming of the uptake rate of the cover cells, not overwhelming the uptake capacity, and it's definitely not killing any sort of cells, not causing any sort of toxicity. Right. And so now I want to just talk about the importance of dose, and I am cognizant of our time. I'll go over this quickly now. The question is, what about other studies, right? We were not fully confident that our experiments would represent a general idea of dose, because we wondered if what we were seeing is just an artifact of the way we made our nanoparticles. Maybe it was an artifact of the way we handled our mice. Maybe it was the way that we were measuring our delivery, right? Maybe there was something going on specifically in our lab. So what we wanted to do was see if there was evidence for this out there in the literature. And so what we find is that exactly the threshold did exist throughout the literature. We used 117 papers from Dr. Wilhelm's paper back in 2016, and we recalculated all of the doses that were reported, and we recalculated them into numbers of nanoparticles. And we were successful in about 40 of these papers, and we got roughly about 60 data points. And what you can see here, and you'll be able to appreciate, is that around 1 trillion, we see this effect that we saw in our experiments to be replicated in the data that was already published. right? And at about 1 trillion, we get this inflection point, and we get this, this increasing function where the nanoparticles delivered better to the tumor as we increase the dose. And so we did some stats on this, and essentially we divided our data points in half. And it turned out that the median dose that was given in these papers was 1.2 trillion. So we divided them into papers that gave less than 1.2 trillion and papers that gave more than 1.2 trillion. And what you can appreciate is that if you gave more dose, you ended up with better delivery. And the question that we asked here is, okay, well, is there some sort of confounding variable here, right? We know that for example, if you give 15 nanometer particles, they're often given in higher amounts simply because of the side effect of normalization. So was this effect that we were seeing, was there a confounding variable that explained why we saw this effect? And what we found is that this threshold was quite robust, no matter how we divided up the data. So we divided the data into different material types, and regardless if it was organic or inorganic, we saw this increase. And if we divided it into these statistical analyses, as you increase the dose, you get increased delivery in both inorganic and organic. And we saw that if you had active targeting versus passive targeting, you get the exact same increase by changing the dose. And this was also independent of size as well, right? So whether or not you gave 15 nanometer particles or you gave 100 nanometer particles, as long as you gave more by number, you saw this increase in dose. And so this really gave us a lot of confidence in our observations in our lab, because this seemed to be generalizable across many different studies and across many different materials and different types of nanoparticles. And so to really assess how confident we are that dose was a parameter, we ran a multivariable analysis on this. And what we found was that dose ended up being the most important and the most confident parameter that we saw. And so on the left here, you'll see a bunch of variables. We had dose, cancer type, active versus passive, size, tumor model cell line, different types of materials. And we looked at how significant they were, statistically significant they were in determining and impacting the delivery to tumors. And what you'll see is that the dose was very significant here with many, many zeros and a 2, and contrasted to a material type that would be rejected in a statistical analysis. You know, things are hard to visualize in a chart. So what I wanted to show you then was a graphical representation of our confidence of this, right? And so we turned this into a word art where the size of the word represents how confident we are that this variable influences delivery. And what you can see here is that we got size is quite important here, but size is not so important when you compare it to cancer type and active targeting. So we're very confident that cancer type and active targeting are significant in the delivery. And this was also found to be statistically significant in Dr. Wilhelm's original analysis, which suggested to us that we were doing the same kind of analysis. Now, all of these pale when you compare dose to these parameters. Right, so dose is really, we're really confident at the end of all of these analyses that dose is a super, super important variable in the delivery of nanoparticles to tumors. And we hope that by the end of this presentation, we're nearing the end now, that you're also convinced that dose is a very important parameter. And so this was all done in mice, a lot of studies about mice. And the question now is, well, what about humans, right? What can we say about humans? So we did a lot of these studies in mice, but we know that mice and humans are different in size, right? And so the mouse is actually, its liver is about 1 gram, and a human liver is about 1.5 kilograms, or 1,500 times larger than the liver of a mouse. Now, if we can make a couple of assumptions, assuming that there's the same cover cell density in a human liver, and there's also, they also have similar phagocytic abilities in the human liver as the mouse, then we can make a quick mass extrapolation here, where our 1 trillion threshold in a mouse could correspond to 1.5 quadrillion in a human. And now, is this our speculation? Or does this have some sort of real clinical consequence? Well, we didn't have the resources or the time to do the experiment. What we did was we started to look at the clinical nanoparticles that are used right now. And we said, are they given above this threshold dose? And indeed, yeah, if you look at some of the more successful nanoparticles, calyx, onivide, myosin, they're all given above this threshold of 1.5 quadrillion, which is represented by this red line. And if you look at some of the high-profile nanoparticles that were in clinical trials but failed and missed their clinical endpoints, things like NK105 and the recent BIND 014 nanoparticles, they were dosed at doses that were below this 1.5 quadrillion number. And so this begs us the question, right? Is this just something that's a coincidence that we're seeing? Or is it that if these two nanoparticles had increased the numbers of nanoparticles to be beyond this line, would they have seen better therapeutic outcomes? And so we're not quite sure at this point. And this really depends on future clinical studies to be able to validate this idea of a nanoparticle threshold in humans. And right now, we really wanted to highlight this because the way that we dose in humans, these drugs, is they're always given by mass of the drug inside with never really any sort of consideration for the numbers or the amounts of nanoparticles that are given. Right? For example, calyx is given at 50 milligrams per meter squared of a human. And they don't talk about the number of nanoparticles that are given. And so maybe in the future, we can start talking about nanoparticles in terms of dose number, as well as the amount of active drug that's given. And so this is the conclusion of the presentation. I leave you with a haiku. You reach dose threshold 2, deliver to the tumors more efficiently. And it's just a simple way to remember what's going on, right? So there's that the dose delivery threshold. Give beyond that, and you'll enable better delivery to tumors. And at the end of this, there are many, many people who are involved in this project, and I just want to send out some brief acknowledgements. Dr. Poon and Dr. Zhang over here had been with me since the beginning of the project. They've been through multiple of these experiments together and have been fantastic teammates and rule of support. Zach Lin and I did the intervital together. He had also done the delivery enhancers. Anthony Tavares, not listed here, but he helped with some of the silica synthesis. Ben Kingston helped us with the 3D imaging. Johnny Zhang over here helped us with the protein corona analysis. And Syed and Presley helped us with some of the 3D imaging as well. Julian Couture-Senecal is not here, but he helped us with some of the in vitro optimization. Fai Song is our lab manager who keeps everything running really smoothly. This is the big boss, Dr. Warren Chan, who has been the major role of support and has been able to lead this project to where it is right now today. And this is the overall Chan lab that makes every day such a pleasure to be working around them. And over here, we've got the Donnelly Center, the CIHR McLaughlin Center, and Vanier for my support roles for funding. And at the end of this, what I wanted to leave you with is a couple more polls here. And I wanted to ask the question here. Stop sharing. And I wanted to ask the question, you know, before this paper and after this paper, did you calculate your doses, and will you calculate your doses by number? And essentially, I guess we're just wondering how much we've convinced you that the dose parameter is an important parameter to use, and will you be using it going on in your studies in the future? And I can't see the responses, so Wilson, whenever you think it's ready, you can feel free to answer that. And I got another question from, while you do that, I'll answer the question from Mohammed. He asked, do you think that different types of nanoparticles will have different thresholds? Yeah, Mohammed, I really, I believe that there will be different thresholds between different nanoparticles. And you can kind of see that already over here. I'm going to go back to this slide. There's quite a spread in the variance of the delivery at each of these different doses, right? Which suggests that by changing the material or the physical chemical properties or the surface of these nanoparticles, you may be able to change the threshold. I truly believe it. We provide 1 trillion as kind of that threshold cutoff for a very broad general idea, but that trillion could be a little bit less or a little bit more depending on the type of nanoparticle. So good question. Shreya asks, Ben, do you think if we found the receptor responsible for this Kupffer cell uptake and blocked it, we could get similar results at lower doses? Yes, I do think so. That would be able, anything that reduces the interaction should be able to get us a different threshold. And secondly, is there any chance we could get even more delivery into the tumor by combining receptor blockade and high dose? Yes, there is. We do have some preliminary evidence that shows that if you do combine the receptor blockade and the high dose, you can get, I think we saw probably an additional 3% delivery into the tumor. So the maximum that we ever saw in the tumor by combining some of these effects was 15% into a tumor. So not 15% ID per G, but absolute 15% of the injected dose into a tumor. Sweet. So I think there are no more questions. I'm going to end the poll now, assuming most people have answered. And I'm going to share results. And wow, this is very stark. I'm happy to see that this variable will be used going forwards. And I hope it does help with a lot of your studies, and I hope it does help with your tumor delivery. And one final question I wanted to ask in this poll is poll number 5. Got 2 questions actually. I don't know if it came up or not.
Okay, I think Sarah asked only drug dose mattered. Also, we pushed higher drug per nanoparticle dose, struggling to make high payload nanoparticles that could also control drug release rate. Sarah, I don't completely understand, but I'm assuming you're saying we also need to focus on increasing the drug per nanoparticle. Yeah, I mean, based on Ben's results, I think if you're able to get more into the tumor, then, and if they're more, oh, okay. Yeah, it makes sense. I think the only thing you might have to worry about is that drug will also go to liver cells if you're doing this. So you have to combine the empty liposome plus loaded liposome. And then that would be another optimization study where you have to figure out what's the right ratio of the decoy and the real, like the drug loaded one, to figure out the maximum efficacy. In terms of biodegradation, I don't think this paper did any degradation because the time points were very short, and gold is kind of hard to follow for degradation, as Wilson knows as well, based on his study. So I think that's a different kind of project where you need to follow it for longer and probably use a particle type that will actually degrade faster. So probably better to do with calyx. There are studies where they can, from our lab, where they've seen at how fast liposomes disintegrate and using imaging techniques, but not for this study.
Sorry for that, guys. I think my internet cut out for a second here.
Yeah.
But I'm back now, and I heard that Shrey has taken care of a lot of these questions. Thanks, Shrey.
Yeah, just the last question, Ben. I think that you can answer a bit better.
Yeah, okay, is there a difference in PK between high and low dose under macrophage depletion? Yes, this is a really great question, Yingyu. And amazing that you asked it. This is a very subtle point, which deserves a little bit of talking about. Yes, there is a difference in PK between high and low dose under macrophage depletion. And so when we depleted the macrophages, there is a prolonged PK, prolonged half-life for high dose. And so this suggested to us that it's not only just the Kupffer cells that are responsible for the uptake. There are also these other cells that are out there. And we know that the RES and the MPS systems are composed of macrophages, they're composed of endothelial cells, and they're composed of these B cells as well. And so by giving a high dose, we are effectively overwhelming all of the receptors in every single one of these cells. right? And so this is why broadly it's more useful to give a high dose than to simply deplete cover cells. Really great.
Yeah, Ben, it's nice talk again. I really enjoyed your PhD defense and your talk today.
Thank you so much, you. I hope that it's
Yeah.
and if we have no more questions, I had one final poll to ask that I didn't get to ask because I got disconnected. And the polls here really are just asking, will you calculate doses for other papers that you've read? And when you're reviewing papers from other groups, will you ask the authors to report their dose by number? So just let us know how you'll approach other papers going forward. And again, Wilson, I'll give you the opportunity here to go versus not go.
We have 7 out of 22 responses. Maybe I'll wait another 10 seconds.
Yeah, I think this this kind of response requires a little bit more thinking. So yeah, and if people say it depends and they want to answer in the chat, that's cool too.
Hi, Ben, just one more quick question.
Yeah.
How long does it take for liver to digest gold nanoparticles in terms of percentage ID? Have you ever done like 1 month value?
Yeah, I mean, we don't believe that the gold is really biodegradable. So it's not going to disappear from the liver, especially not within 1 month. We have a couple of studies that we've done, and Dr. Poon has meticulously analyzed how gold can be removed from a liver. And you can read that in his ACS nano paper. But essentially, he found that the body can clear a maximum of maybe about 10% of the gold that you inject into it, and the rest of it just kind of stays there. And we're not aware of any enzymes or any compounds that can degrade gold from the body, and so it kind of just hangs out. And so this is part of the reason why we're doing our long-term toxicity study, where we injected our animals 8 months ago and are now kind of waiting to see what ends up happening, right? Is there chronic changes? Yes, maybe. There was that PNA study that came out, I think, last year.
Regarding the
months. There
biodegradation, right?
Yeah, and there's also the PNA study that showed some sort of degradation. Yes, I agree. So we're going to be.
Yeah, but that happened to small gold nanoparticles. They claim it's methylthionine, right?
Methylthionine.
I agree with Yingyu. I think if you're going to have degradation, I think it's going to be for really small gold nanoparticles.
Yep.
Like even if for the small nanoparticles in their TEM images, it was like very subtle difference. So if you have a 50 nanometer gold nanoparticle, especially that's
there's a different story. Yeah,
exactly.
The Peanuts paper, they did 4 nanometers and 22 nanometers.
Yeah,
right?
Exactly. Yeah. But I'm gonna end the polling now.
Cool. All right. Cool. So this is exciting. Yeah, I'm glad that people will try to calculate the doses for the other papers that you read. And I'm glad that you'll suggest that people report doses by number. Hopefully we can establish a dose metric that's used across the field, and that will help us move forward in our ability to deliver nanoparticles much better in the future. But thank you everyone for coming. This is a Saturday. I'm so happy that you guys spent an hour and a half with us doing academic stuff on your weekends. I'm really grateful for your attention. Hope to talk to everyone soon. Thanks for the great questions.
Thanks, Ben. This was a really awesome presentation, and I really appreciate how you made it very understandable too. Thank you.
Cool. I'm glad. Thank you, everyone.
Thank you, everyone. We will have our next session in 2 weeks. And I think Wilson already has everyone on the mailing list. So be on the lookout for that, and we'll send it again in 2 weeks. And we're trying to diversify our topics this season. So keep an eye out for it, and we will be with you in 2 weeks again.
And can I ask a quick question?
Yes, Stefan, what's up?
So thank you again, excellent presentation. If those gold nanoparticles are remaining within like biological systems like the liver and stuff, if you wanted to give like another drug, would those, like the liver already be saturated, so wouldn't it be as much of a challenge?
Yeah, so this is an interesting question. We know that at the low doses, the liver itself, if you get the repeated doses, the liver never really saturates its capacity, and it can continue to take up more and more and more. The next question then is, if we give a super high saturating dose, and then we wait for it to all clear, and then we give a second high saturating dose, what ends up happening? We never got around to doing that experiment, but it's a very good question. And I know you're starting in our lab soon, so maybe it's something you might want to explore.
Yeah, thank you so much.
Yeah, really good question. Thanks.
Thank you all.